Hi, my dear students. How are you all doing? All of you, all my students are doing great. Yes, yes. Then it is good. A very warm welcome, guys. Here, this is a series. The summary series is going on, and today I'll be talking about the neural control and coordination. So this chapter we will be discussing in a short way. I'll be explaining you all the concept in a brief. So let's start the session. Now these sessions, they are helpful for you for a last minute revision where we are explaining everything as per your exam, your from your examination point of view. So let's start. So neural control and coordination we will be discussing. Okay. So what are the further subtopics of this chapter? Further subtopics of this chapter is the neuron. So first, that is the neuron. So one second. Yes. So first I will be talking about the nervous system. We'll wait for some time so that our screen should be back. So we will be talking about the neuron. Yes, now it is back. So first, guys, we will be talking about the nervous system. Nervous system. We have neurons, we have ganglia, we have a central nervous system. What is that? What is the peripheral nervous system? Everything we will be discussing. Then I'll be talking about the basic unit of our nervous system, which is, what is that? That is a neuron. The structure and the function of neuron. Then we will be talking about the conduction of nerve impulse. Conduction of nerve impulse. Right, this we have to discuss. Then the parts of the central nervous system. We will not be discussing in detail the spinal cord. We will be only talking about the parts of the brain. Forebrain, midbrain, hindbrain, what does it control? Everything as per your book, as per your reference book, your NCRT, we will be discussing. So let's start. What is a neural system? The neural system of all animal is composed of highly specialized cells which are termed as neuron. And what is the function of neuron? Neurons, they detect, they receive, and they transmit the different kind of stimuli. We are able to sense anything just because we have a neurons present in our body. Now neurons are not long, they are of a normal size, they have a very normal size, but then how do they transmit information? So they basically transmit information from one neuron to another. I'll give you a very good example. Suppose this is a hot surface. Consider that this board is a hot surface, and I by accidentally I touch this surface. What will happen? Within a few seconds, in fact within few milliseconds, I will remove my hand, because the signal has been sent to the central nervous system, and central nervous system will cause the contraction of these muscles, due to which my hand will be removed from this hot surface. So that communication towards the brain, and then there is a motor sensation towards the muscle. So all of this, they are under the control of neurons. So here are the neurons which helps in this type of communication. So that is the reason it is written as detection. We detect hot, cold, and we send the signal to the central nervous system. So they receive, they transmit, everything is because of the neurons. Yes, clear? Okay. So first of all, see, now I am explaining you in a short way everything. I'm writing, I'm giving you some short notes as well. So let's talk about the nervous system. Nervous system. Let's talk about the nervous system. We all know we are having a central nervous system, which I'm making with a simple diagram. What is this? The structure which I have drawn above, what is this? This is a brain. And what about this long structure? This I'm labeling as the spinal cord. Now what does a brain and the spinal cord form? Brain and the spinal cord forms the central nervous system. What does it form, guys? Central nervous. What does it form? Central nervous system. Am I right? Central nervous system. So it forms the central nervous system. Now, these brain and the spinal cord, they send signal to involuntary structure. to the involuntary organs? On the contrary, not only the involuntary organ, they send signal to the voluntary organs also. Voluntary organs also. Right? They are going to send signal in both the directions. Now, just imagine when they send the signal to the involuntary structure, and definitely this type of communication is C, right? This comes under the autonomous nervous system. What do you mean by this? Autonomous nervous system. Now, whereas this type of communication between the voluntary organs to the brain and the spinal cord, this is termed as somatic nervous system. Somatic nervous or neural system, whatever you want to write, you can write down. System. Clear? Somatic nervous system and autonomous nervous system. So there is a two-way type of communication. The brain and the spinal cord, voluntary and the involuntary organs. Now, you will see that some nerves, they go to the involuntary and voluntary organ from the brain. So these nerves, they are termed as cranial nerves. So what is the name given to them, guys? These are your, here I am labeling, these are your cranial nerve. How many cranial nerves do we have? 12 pairs. Characteristic feature of mammals. 12 pairs. And when it goes from the spinal cord, that will be termed as the spinal nerves. Spinal nerves. How many? 31 pairs. 31 pairs. So when I'm saying the somatic nervous system or the autonomous nervous system and somatic nervous system, they both are the part of the peripheral nervous system. Peripheral nervous system. Now listen to this point, guys, very, very, very carefully. There is a central nervous system and there is a peripheral nervous system. Peripheral nervous system include your somatic nervous system. If we talk about the voluntary organ, when we say the autonomous, that means we are talking about the involuntary organs. So the points are clear, guys. Different names related to the voluntary and involuntary structure. Now again, listen very carefully. Now, suppose this is a central nervous system, right? Now in this central nervous system, from the, let's say, from the your receptors, these are your receptors, right? Now receptors sense the signal, and this signal will go to the central nervous system. Right? Receptor, they will sense something. Let's say they sensed heat, hot condition, heat. Signal will go to the central nervous system, and these type of neurons, nerves are what? Nerves are the collection of neurons, right? So when I'm saying the neurons are there, so neuron is a single unit. When I'm saying nerves means collection, right? Those nerves which carries the signal from receptor to the central nervous system, they are termed as sensory nerves. They are the sensory nerves. Now, central nervous system will send the signal to the effector structure. Now, this effector structure can be your muscles, can be anything, right? Now, this is termed as the motor neurons. So hope this revision is clear. What is nervous system? What is central nervous system? Peripheral nervous system, receptors, etc. and etc. Right? Now let's move on to the structure of neuron. Structure of neuron. See, ultimately whatever you are studying, that should remain in your mind for long time. That is my motive is. See, this is how a neuron look like. Here you will see myelin sheath is present. What you will see is myelin sheath is present, right? And these myelin sheath is formed by the Schwann cells. I'm making a modified diagram of your NCRT for the clear explanation of each and every topic. Now let's talk about this neuron. Here in the neuron, you will see there is a cell body present. What is this? This is a cell body. What is this? The whole structure is cell body. In the cell body, you will see a nucleus is present. Can you see a nucleus? So yes, what is this? This is a nucleus. Cell, because it's a cell, so definitely they will be having living cells. So definitely the nucleus should be present. Now here some granules are present, and these granules, they are derived from ribosome and rough endoplasmic reticulum. They are termed as the Niesel's granule. Right? They are formed by the rough endoplasmic reticulum plus ribosomes. Very important question. Right? Now in the cell body, you will see some processes, and these processes, they are termed as dendrite. These are termed as dendrite. Some processes, they are coming out, dendrite. Now this particular white structure which you can see, this is termed as the axon. What is this? Axon. So one process is long, which is a axon. Ultimately, these are the axon terminal. What do we call this? Axon terminal. And these are termed as axon knob. Now signal comes from dendrite, and this is how it travel, right? This is how it travel. There are some receptors present on this structure. See, the receptors, they are present over here, right? They will sense the change, and they are going to transmit the information towards the axon terminal. Now as I have told you, now this is termed as a myelin sheath. It's not necessary that myelin sheath should be present. The neuron can be myelinated or unmyelinated. Both things are possible. Here, what do I mean by this? These are the Schwann cell. In case of central nervous system, this is formed by the oligodendrocytes. Right? Structure is clear. Now this is termed as axon hillock. Axon hillock. So this is the difference. Clear? Now what happens is, here they will sense the signal. Signal will be transmitted towards this direction, and here you will see the neurotransmitters are present in these vesicles. Neurotransmitters are present, and these neurotransmitters, they show the exocytosis, and that come in this space, which will go and bind to the receptors of the next neuron. So this is how the communication occurs. So this is a structure of a neuron. Now let's talk about the types of nerve fiber. Now these nerve fiber are of two types, right? Now one is a myelinated axon. Myelinated means myelin sheath is present. That means enveloped with the Schwann cell that form a myelin sheath around the axon. If myelin sheath is present, so that means the myelinated one. Now next is non-myelinated. Non-myelinated means Schwann cells are present, but they are not forming the myelin sheath. They are found in the peripheral nervous system. Now the white colored area formed by the myelinated nerve fiber is called white matter. So what do we call that? The white matter. The gray colored area without the myelin sheath is called as gray matter. Now found in the spinal and the cranial nerves. In the spinal as well as cranial nerves, you will find this. Whereas what about non-myelinated? Non-myelinated are always present in autonomous neural system. They are found in the autonomous and somatic neural system. Clear? Is this point clear? No. These neurons, they are of various types. Now, if I talk about this neuron, this structure we have done, where we have seen there is a cell body and there is one dendrite, one axon present. This is termed as a multipolar neuron, which is present throughout the body. This type of neurons, they are present. If I am saying the neuron is like this, neuron is like this, here you will see the dendrites are there and one axon is present. So you will see one process is coming here and another process is here. So this is termed as a bipolar neuron, which is present in the nasal epithelium as well as in retina of the eye. Pseudo unipolar neurons. See, it looks like bipolar, but they are not bipolar. Here what happens is the dendrite, they further bifurcates into two. See, this is the one process which comes from the cell body, which further bifurcates into two. So this is termed as pseudo unipolar. This is seen in the dorsal root ganglion. Unipolar looks like this. This is seen in case of the hydra, or in fact the apolar are also there, which is seen in case of the hydra. In fact, in the embryonic stages also the unipolar neurons are present. Now question arises, how there is generation and conduction of nerve impulse. Let's do this revision at a very fast rate, at a very fast pace. So this topic gives us an idea that how there is conduction of nerve impulse. See, I told you here conduction of nerve impulse, that means from the dendrite towards the axon, right, towards the axon terminal, there is a conduction of nerve impulse. So how does it occur? Let's have a look. Now when it is, the neuron is in resting stage. Where is neuron? Neuron is in resting stage. Do not read this. I'm going to make you, this is a very important thing which I have done, that is I have made a chart so that you can have an easy understanding of this topic. When it is not conducting any impulses at rest, when neuron is at rest, neuron is resting. So this is termed as a resting state, right? Now here at resting stage also, you should have in that idea that inside the neuron, K plus ion concentration is more and outside sodium ion concentration is more. That means in the outside the neuron, outside the cell, the sodium ion concentration is more. Axonomal membrane is permeable to K plus, nearly impermeable to sodium and negatively charged protein. Now what they are saying is, this is a neuron, right? Now they are saying they are permeable to K plus. So K plus leakage channels are present, which due to which the K plus is more towards inside, and inside there are some negative charge protein present. Negative charge protein, right? And here they are impermeable to sodium, so the sodium is present more on outer side. Here the negative charge ion is more, so inside it is negative, outside it is positive. Towards the extracellular region it is positive. Now listen, nearly impermeable to sodium ion and negatively charged protein. Inside the axon, high concentration of K plus, axon inside, high concentration of K plus. Negative charge protein, low concentration of sodium ion, fluid outside the axon contain low concentration of potassium and a high concentration of sodium. So this point I have explained you with the help of this diagram. Now what will happen? This will result in the formation of a concentration gradient. What will happen? Concentration gradient. Outside positive, inside negative. Maintained by the sodium potassium pump, which transport 3 sodium out for 2 potassium ion inside the cell. Axonomal membrane in this case is polarized, outside positive, inside negative. The electrical potential difference across the plasma membrane is termed as a resting potential. What do we call this? Resting potential. So here also potential is there. So just imagine the situation. Outside it is, see, consider that I'm a neuron. Outside me, sodium ion is more. Inside me, potassium ion is more. How it is being maintained? Because our axon, our neuron, they are pushing sodium out and they are bringing its potassium ion inside. But inside we are negative. The reason is the presence of the negative charged ion. Inside negative, outside we are positive. Now, what happens over here? Now, when there is a start of impulse, that means now the conduction has to start. At this time, what happens is a stimulus is applied at the site A in a polarized membrane. Stimulus came. That means now the potential has to change. right? Axonal membrane permeable to sodium ion and impermeable to potassium ion. Now the everything will change. It will become permeable to sodium ion, impermeable to potassium ion. Rapid influx of sodium ion. Sodium ion will come dhadale se inside the cell. Axonal membrane outside negative, inside positive. Clear? Now, due to this, what happens is polarity of membrane at the A site is thus reversed. Now, if we have a particular site here at the A site, suppose the impulse will come over here, right? What will happen? Sodium ion will gush into the neuron, due to which inside it become positive and outside negative. Polarity of membrane at A site is reversed, hence this condition will be termed as depolarized. The electrical impulse difference across the plasma membrane at this site where the stimulus is applied is called as action potential. Now this is termed as an action potential. Now which is in fact termed as a nerve impulse. What do we call this? Nerve impulse, right? Now at sites immediately ahead the axon, that is a B region, membrane has positive on outer and negative on inner. So this is one part. So if you look at the part, this another part of the same neuron, this is a B part. But in B part, outside it is positive and inside it is negative. Clear? So that means action potential has reached at the A region, not at the B region. Clear? Now, the current flows on the inner surface from A to B, outer surface current flow from the B to A. Now, what they are saying is, buddies, so here current, because this is a negative charge, here it is the positive charge, you remember. So, what will happen, buddies? Current will flow from A to B, outside to inside, from inside like this. So, this is a current flow will occur, right? To complete the circuit. Hence, the polarity of the site is reversed and action potential is generated at B. Now, the same thing will happen at the B. At the B site, what will happen? Sodium ion channel will open, sodium ion will come inside become negative, right? Now, the same thing which happened at the A site, now that thing is going to happen at the B site, right? The impulse generated at the A site arrives at the B. Sequence is repeated along with the length of axon and impulse is conducted. So first I am in a resting state. Inside I am negative, outside I am positive. Now consider that this is a A region, right? This is a A region. Here, as the impulse will come, so there will be opening of the sodium ion channel. Inside it will become positive, outside negative. This is the region number B. Region number A, both are adjacent to each other. They will form a current. There will be flow of the circuit outside from the A to B and inside from the B to A. A circle formation will occur, and because of this circle formation, this signal will transfer to this signal. Here the sodium ion channel will open. Now this area will become depolarized. Initially it was in resting state. Now it will become depolarized. Now again repolarization. Now A region has to repolarize it. What happens is the rise in the stimulus induced the permeability of sodium ion is extremely short-lived. It is quickly followed by the rise in permeability of the potassium ion channel. Now this area, sodium ion channel will open. This happens only for a few duration of time. Now the potassium ion channel will open over here. As the potassium ion channel will open, potassium ion will gush into the cell. As they will gush into the cell, now the potential will be changed. Now listen very carefully. Within a fraction of second, K plus diffuse outside the membrane and restore the resting potential of the membrane at the site of excitation. Fiber become more responsive to further stimulation. Sorry, there is a thing. Now here what happens is, this was the A region and this is the B region. Two different regions are there, and in this A region, first there was the opening of the sodium ion channel. Sodium ion, they were coming inside. Now there is opening of the potassium ion channel. Potassium ion will go out. As potassium ion will go out, inside they will again become negative, outside they will become positive. So this is how the things happens. Now the signal has reached over here. Here the sodium ion channel, they are open. Adjacent to this, there is a C region. In the C region, again there is an electric circuit formation. There will be opening of the sodium ion channel. At the B region, what happens is there will be opening of the potassium ion channel. Potassium ion will go out, inside become negative again, outside become positive again. So this keeps on traveling. right? Opening of the potassium ion channel is termed as a repolarization. See, see, at a resting membrane potential, see, resting membrane potential, the potential is -70 only. Later on, as there is generation of impulse, there is opening of the sodium ion channel, the impulse become +30. It becomes +30. Now it has reached to the +30. Now once what will happen at the region number 3, there will be opening of the potassium ion channel. Potassium ion channel, because of the more opening of the potassium ion channel, it will become more negative and then it will come back. See, I'll tell you how it happens. First, this is a first potential. Here everything is being maintained by the sodium potassium pump. Who maintains this? Sodium potassium pump. Sodium potassium pump maintains that inside it should be negative and outside it should be positive. Here the potential is -70 millivolt. Now when the impulse come, what happens is there is opening of the sodium ion channel, due to which outside sodium comes inside the neuron, right? If they will come inside the neuron, so inside it will become positive. It will reaches to the +30. Depolarization, voltage gated sodium ion channel will open. Now what happens is, because it has reached to a particular destination, now it has to come back, because sodium ion channel, they are short lived. They open only for a short lived because they are voltage gated. Now what happens is there is opening of the potassium ion channel. Because of the potassium ion channel, potassium ion will go out. As the potassium ion will go out, inside becomes negative, which was positive earlier. Now it will become negative again. Now as it will become negative, it will come back to the normal value of -70, right? -70. Now for a few time, for some millisecond, the potassium ion channel remains open, due to which the resting membrane potential becomes more negative. This is termed as hyperpolarization stage. Now this stage, which you can see over here, is the hyperpolarization stage. Hyperpolarization state. Hyperpolarized this condition is, right? And again it will come back, but the sodium potassium channel will maintain this. There will be again it will come back to the -70 millivolts. So -70 +30 and again. So this is how it happens. So simultaneously, guys, you all are having your notes ready also. So this is a diagram given in your NCERT. Now first what happens is, if one part is depolarized, that means impulse has been generated. Another part is resting state. Now what happens is, it will become repolarized. The next part will become depolarized. Now this repolarization will come back to the resting stage. Depolarization will form the repolarization. There is a transmission of this green part from one part of the neuron to another by the formation of that electric circuit. You remember, A to B and B to A. So this is how it happens. Now my next thing is synapse. So what is a synapse? Synapse is basically a communication. It is a kind of communication between the two neurons. So this is termed as a synapse. So how does it occur? Here it occurs basically between the axon of presynaptic neuron, presynaptic neuron with dendrites of postsynaptic neuron. So you will see here a space is present, and that space is termed as synaptic cleft. So here you will see, here this is a presynaptic neuron, second one is a postsynaptic neuron. Now once the signal will reach up to here, now signal has to transfer from this neuron to another neuron. There is always a presence of a space between them, which is termed as the synaptic cleft. What is this? This is the synaptic cleft. This space is termed as a synaptic cleft is present. Now as the signal will reach over here, right, here what happens, buddies, here at this particular region there is influx of the calcium ion channel. There is opening of the calcium ion channel, due to which here the neurotransmitters are present in the vesicles. These neurotransmitter will go and they will show the exocytosis. Neurotransmitter will come in this region. Here the neurotransmitters are present, right? neurotransmitter will come here in the synaptic cleft region. This is a cleft. They will come over here, right? Now here, buddies, you will see the dendrites, the receptors are present. These neurotransmitter, they will go and they will bind to the receptors. There are various kind of neurotransmitter that we have learned in the session, which is your GABA, acetylcholine, glycine, aspartame. So many types are there, and they help in this kind of conduction. There are some type of neurotransmitter which are excitatory type, some are inhibitory type. So this is how the communication happens. So exocytosis, they are going to communicate. This type of synapse is termed as a chemical synapse. This is a chemical synapse. But when the distance is very, very, very less, there is direct transfer of these signals from one to another. Direct transfer is also possible. Now that signal transfer is termed as an electrical synapse. But here, this is the chemical synapse, right? This is a synapse. See, look at this diagram. This is the axon terminal. You will see the vesicles are there filled with a neurotransmitter. Because of the calcium ion influx, there will be exocytosis of this synaptic vesicle, and these synaptic vesicles, they are going to fuse with the membrane, due to which the they will come into this space. When they will come into this space, they are going to bind to the receptors present on the dendrite. So this is how the communication happens. Clear? Now in this case, many students, they keep on asking me, ma'am, what about this neurotransmitter? What happens to this neurotransmitter? Do they enter into the next cell? No, they do not enter into the next cell. They, in fact, they open the ligand-gated sodium ion channel. Because of the opening of the sodium ligand-gated sodium ion channel, so from the resting state, the neuron comes into the depolarized state, right? Now this depolarized state will cause the next region to become depolarized. But these neurotransmitter, they are recycled again. These neurotransmitter will be further broken down. For example, acetylcholine will be broken down by the acetylcholine esterase into acetyl plus choline. Choline again will be recycled by acetyl coenzyme and so on, and ultimately they will form the acetylcholine. So there is always a recycling. Our body do not waste good things. Our whole process, our whole system is super duper well designed. Now, let's move on to the next part of this chapter. What is that? That is a central nervous system. So what is a central nervous system? Central nervous system is a major processing unit which controls the major activities of our body. Whether it is related to your speech, communication, memory, recognition, sexual desire, hunger, everything is under the control of the central nervous system. So in central nervous system, there are two different parts. One is termed as the brain, and second is termed as the spinal cord. Two different parts are there, brain and the spinal cord, right? So brain part is the one which we will be discussing in detail. For example, the brain is the central information processing organ of our body and act as command and the control system. It controls various activities of our body. Now, if you look at the brain is covered by different menings. The outer cover is termed as a durameter, then the arachnoid, and then the pyometer. Three different layers are present: durameter, arachnoid, and pyometer. Between the arachnoid and the pyometer, there is a presence of the subarachnoid space. Subarachnoid space, and in the subarachnoid space, CSF is present. What is CSF? Cerebrospinal fluid is present. Outer is a durameter, arachnoid, and the pyometer. These are the three layers which are present. You have to know that which one is vascularized, which one is not. So this we will be discussing in the detailed session. So if you will go further, see, first is a cranium, which is a brain box present. Below this, you will find these three layers are present, which are the DAP. And below this particular thing is present, which you can call it as a brain. Right? So here you will see the whole brain is divided into three different parts. The first part is termed as a forebrain. This is a forebrain. Second is a midbrain. Now next is a hindbrain present. Now forebrain, the further different parts are there. So forebrain, major area is occupied by the cerebrum, where the folds are present, the gyri and the sulci are present. We have a two cerebral hemisphere, right cerebral hemisphere and left cerebral hemisphere. And these cerebral hemisphere, they are connected by a tuft of neuron, and that tuft of neuron is termed as the corpus callosum, right? This is the one which connects. Now, other part of the forebrains are your thalamus, epithalamus, hypothalamus. These structures, they are also present. Other structures are also there, which we will be discussing, such as your amygdala is there. The optic lobes are also present. So that we will be discussing later on. Thalamus and hypothalamus is termed, thalamus is a relay center of the body. Whatever communication that goes to the cerebrum part, that passes with the help of the thalamus. So thalamus is that relay center. Next is a hypothalamus. Hypothalamus is that structure which controls not only the nervous part, it controls the endocrine part also. You know, it secretes hormone also, which goes directly to the posterior pituitary, and they are secreted from them. Hypothalamus also secretes some hormone which affect the secretion of the anterior pituitary. Next is a midbrain present, right? Here the colliculi are present, two superior colliculi, two inferior colliculi, that I'll also explain you. Hindbrain, the three parts, the pons, cerebellum and the medulla, further parts are there, which will further communicate to the spinal cord. Chalo. First, let's talk about the forebrain. So forebrain, here it consists of the cerebral, the thalamus and hypothalamus part. This is what they are saying. And cerebrum is considered as the largest part of the brain. And if you took a look at the cerebrum, here the structure look like this. So here you will see the folds are present. These are termed as a gyri. And the depression is termed as a sulci. This increases the surface area so that they can accommodate more and more neuron. Here, the outside part is grey in appearance. Why? Because the axon cell body, the neuron cell body is present. So if you zoom this structure here, I'll show you. Wait guys. If you zoom this structure like this, right? You will see the neurons, they are present. Right, this is how they are present. So here, because of the presence of cell body, so they appear grey in color. So the cortex area is grey in color, whereas if you talk about the medulla part, they are the white in color because the myelin sheath is present. Right, white myelin sheath is present. So the cerebrum has a various part. This is a frontal part, this is the parietal part, this is a temporal part, and this is the occipital part. These are the different lobes. Two frontal, two parietal, two temporal and two occipital part are there, and each part they control the different the actions of the body, which we will see in the detailed session. Now, a deep cleft is present in the cerebral longitudinally into two equal half. So if you look at the cerebral hemisphere, so this is how a cerebral hemisphere look like. Here the deep cleft is present which divides right cerebral hemisphere and the left cerebral hemisphere. Now they are connected by a track of the nerve fiber, which I have explained you, termed as a corpus callosum. Cerebral cortex is a region where the grey matter due to greyish in appearance, which is present towards the outer side. Now cortex region contains the motor area, sensory area, and the large part is the association area. Right, now if you look at, I'll show you, wait. So if you look at, this is the cerebral cortex, some sensory area is present, some motor area is present, but the major part is the association area. Major part is the association area, and this association area is that area which controls the memory, which controls the communication. So intersensory function. Intersensory, that means at a one particular time you are smelling your food also, you're tasting your food also, so that is possible because of the association area. right? So various areas they are present. Cortex contain motor areas, sensory areas, and large region that are neither clearly sensory nor motor. These are termed as association area, responsible for intersensory functions, memory and communications. Direct line, guys, NCRT. Oof. Now, fibers of the tracts are covered with myelin sheath, which constitute the inner part of the cerebral hemisphere. They give an opaque white appearance. Why? Because of the myelin sheath. So they are termed as a white matter. Now, next part is a thalamus. Thalamus is termed as a relay center. What do we call this? My very important question. This is a relay center. Whatever information goes to the cerebrum for the processing, that transfer through the thalamus, right? So this is a major coordinating center for motor and the sensory signaling. Now next is a hypothalamus. So what is a hypothalamus? Hypothalamus lies at the base of the thalamus and it controls the body temperature, right? This is the one which is responsible for eating, for drinking, hypothalamus. Next, very important, limbic system. So what do you mean by limbic system? Limbic system means your hippocampus, amygdala, right? Hippocampus, when we talk about the hippocampus, amygdala and the thalamus. All these three structures, they form the limbic system. But when we add here the hypothalamus, we call this system as, what do we call this system? We call this system as emotional brain. Why emotional brain? Because now it controls the various emotions. What are different emotions? Such as sexual drive, your eating, your thinking, your communication, everything will be controlled by the heart of hippo. Right? Now the next one is midbrain. Midbrain is located between the thalamus and hypothalamus of forebrain and the pons of the hindbrain. First part of the hindbrain is pons and the last part is thalamus and hypothalamus. So in between them you will find the midbrain, comprised of the cerebellum, aqueduct and corpora quadrigemina. Okay. So corpora quadrigemina, here you will find two superior colliculi are there. And two inferior colliculi are there. So if I talk about, I'll explain you cerebral aqueduct also, just wait for some time. Superior colliculi and inferior colliculi. Superior colliculi are the one, so here quadrigemina, 4 lobes are present, 2 are present at the upper position, 2 are present at the lower position. Superior and inferior. Superior colliculi, they are important for the visual reflex and they are the one which are responsible for the auditory reflex. Now suppose there is a light coming in your room and your room is, very dark, right? There's no light in your room and from a window, from a place, some light is coming. You are going to look at that thing. So that is termed as a visual reflex, right? Suppose you're going in a lane and suddenly you will realize that some light is coming from the background. What you will see? You will turn your head, right? This is termed as a visual reflex. Now the next thing is the auditory reflex. Auditory reflex, that means something is at your back and someone is calling your name in a dark, in a dark lane. It's the mid of the it's the midnight. Someone is calling you from the background. Javi, something like this. You'll be scared. You will turn your head. For the auditory, just to you want to listen to that voice, you turned your head. That is the auditory reflex. Next is the cerebral aqueduct. Cerebral aqueduct is present between the ventricles of the brain, where you will see the third ventricle is connected to the fourth ventricle with the help of a duct, which is termed as a cerebral aqueduct, which connects the third ventricle with the fourth ventricle, the last ventricle. That is termed as a cerebral aqueduct, and cerebral aqueduct surrounds the midbrain. So in cerebral aqueduct you will see the CSF is filled, right? Okay. Now it coordinates the movement, head, eyes and trunk in response to the visual and the auditory stimuli. Next is a hindbrain. It consists of pons, cerebellum and the medulla. First part is the pons. The pons is a relay nerve impulse to the cerebellum, cortex, and consists of numerous fibers that interconnect different region of the brain. Different region of the brain, they are connected to the body through the pons, right? Of the different region of the brain is connected with the help of a pons. Now, you remember in breathing and exchange of gases, we have talked about pons. So pons is having pneumotexis center. So it regulates the breathing. You remember we talked about that's the switch off center for inspiration. So yes, here it is present. Next is the medulla oblongata. Yes, breathing with medulla oblongata. It is connects with the spinal cord. So here it is present. In South Indian, in many movies, I'm a very big fan of the South Indian movies. So in movies, what happens whenever there is an action scene, whenever a villain hits here, the person becomes gone. So person will no longer be in the conscious state. So what happens is, it is only because this connection, that is a medulla oblongata, is being affected, which connects the brain to the spinal cord. So this is a quite sensitive area. One should not hit over here, because here the medulla oblongata is present, which is a communication between the brain and the spinal cord. Autonomic center for the regulation of the physical functions such as the cardiovascular and the digestive activity. You remember where is a vomiting center present? Medulla oblongata. To talk about the cardiovascular, that means the vagus nerve that comes from the medulla oblongata only, which reduces the the impulse generated by the SA node from 100 to 70. You remember that also we have discussed in the session. Now, next is a cerebellum. Now the cerebellum is the second largest part of the brain, not the first, the second largest part. It is again convoluted, just like the cerebrum. Very convoluted, provide the additional space for many neurons, smooths and coordinate contraction of the skeletal muscle, regulate the posture and balance. This is what the cerebellum is, right? So today we have talked about your nervous control and coordination in a brief way. I have given you a quick revision of the whole chapter. If you want the detailed one, any topic, please let me know in the comment section. Till then guys, have a nice day. See you in the next class. Take care, keep studying, keep yourself safe in this time. Bye bye and all the best for the examination. Thank you so much.